my name is Aina. Maybe you know me from other Instagram's live TV that I've done here. I'm one of the embryologists of Repro Clinic, and today I'm just going to answer a few questions that we've received from you guys about the lab, and embryos, and eggs, and a bit of everything. If you have any questions while I'm speaking, you can right there. I think I'll read it and I'll try to do my best. It's a bit odd to just be speaking to a phone. I have here some of the questions that you guys sent, so I'm just going to read them and try to answer in the easiest way that everyone can understand. All right. First question that we've received is, could you explain how the sperm and the eggs are freezed in the lab? All right. The process between is different from sperm and from eggs, but more or less the technique, well, how it works is kind of the same. At the end, we work with cryoprotectors, and that's a media that what actually does, it just protect the proper cell of the freezing that we are going to do. Because, for example, first of all, I'm going to explain eggs and then sperm. Eggs is quite similar with embryos. It's not exactly the same process, but actually it's kind of the same at the end. It's not like the same steps that we do. Like timings are a bit different, but the process it's kind of the same. We work with cryoprotectors, and first of all, you need to put those eggs in cryoprotector, and you need to be adding cryoprotector every like 3 minutes. We work with Kitasato, and what it does, the proper egg will like take the water that it has inside out and will accept the cryoprotector, and that will help because if not, if you freeze the egg, we freeze with liquid nitrogen, so it's minus 195 degrees. So if there's water inside the egg, it will form like crystals, like the snow. So the proper cell will be degenerated. So you need to remove all the water in terms of the egg to be able to be frozen. Okay, so we put the eggs in a media and we keep adding cryoprotector like every 3 minutes. So the concentration is getting higher and higher, because if you just do it all together, it's not good for the egg. So it needs to be adapted. Think that it's just one cell what you're freezing. Okay, so after that, you will actually take the egg, put it in the cryotop. Today I don't have it, but in another Instagram TV that I did about egg freezing, I show it. It's like a little bar like this that has a on the edge. It has like a kind of plastic, and it's quite of not rigid. So over there you put the egg with a little bit of media, and you put it directly to the liquid nitrogen. And then once it's inside, you can never move it. All right. So it needs to be inside the liquid nitrogen until you actually are going to thaw. And the process of thawing is the other way around. So you will remove the cryoprotector and put back the water to the egg. All right. With the sperm, the process is a bit easier, and you don't need to do actually anything to the sample. So it will be the proper sample. You don't need to do any gradients or anything. You might do if there's need, but if not, you can actually freeze it as it is. Okay, and you just mix the sample with cryoprotector, and then after that you will put it in another dispositive. It's like I don't know the name in English. It's called pajuela in Spanish, but I don't really know the name in English. It's like this, kind of this big, and inside it's like a plastic bar, and then you will close it this way and that way, and that's it. And when you're going to thaw it, you will cut it from one side and the other one, and that's it. And it's quite easier from the sperm to be frozen than the eggs, because obviously the egg is just one cell, so if the sperm, you won't expect all the spermatozoids to survive, but you will actually have much more sperm than eggs. So after the thawing, if you just have half of them that survive the freezing, that's alright, and you will have enough. So that's why actually the process is not that complicated. Our second question is the embryos, the egg, or the sperm are preserved in a single pack or in small groups. All right. Normally, embryos are frozen in alone, like single pack, because then when there's a cryo transfer, you can actually decide if you want to thaw one embryo or two, based on if you're going to do a single transfer or double transfer. All right. Normally, we just freeze one by one, and then the patient will have the chance to decide, and that's it. With eggs, we normally freeze in packs. We actually do in packs of two, and then maybe three. It depends on the number of eggs that we have. So if it's like 10 eggs, we will freeze 5 cryotubs with 2 eggs in each. And then if you have 11, you will have 4 cryotubs with 2 eggs in each, and then the other one of 3, because you're not going to freeze only 1 egg. That's because we never expect to thaw actually 1 egg for a cycle. It wouldn't actually make that much sense to just thaw 1 egg and actually thaw and fertilize only 1 egg. So at least these 2. You could actually do 1 if there's the need of someone for any beliefs or wherever they would just want 1 to be fertilized. But our normal procedure is 2. But there's places that they freeze in packs of 4 or 5, so you can actually do. I have never seen more than 5 actually in one cryotop, but yeah, we do groups of 2. And with the sperm, you actually freeze like the sample in each dispositive. You can have like 0.5 milliliters. So normally, if it's just for someone who's freezing for using it afterwards treatment, we would freeze like 8 maybe or 5. It depends on the volume that the sample has. But if it's for like keeping the sample for something like preservation, then you will freeze all the volume. But for all the dispositive that you will have, you will only thaw 1 normally for each treatment. It's just if the sample is really, really bad that you would remove, you will use more than 1 dispositive. Then what else? Is there a time limit on how long embryos gametes can remain frozen, like duration of storage? No, there's no limit. Once it's frozen, it can be frozen like for years and years. The only limit actually would be like the age limit of the treatment that you can have, but it would be like 50 years, I think here, maybe 51 if there's a bit of exceptions, but normally it's like around 50 years. But there's no actually time of the embryos or eggs or sperm of being stored. Once they are stored in liquid nitrogen, it's like if time stop, and that's it. It doesn't matter if it's 1 year, 2 years, or 10 years. It would be exactly the same. All right, so there's no worry for that. And then next question, can you describe the process of fecundation, like the fertilization that we do in the lab? There's actually in treatments, I would say like there's 3 treatments of fertilization that you can have. And there's insemination, there's IVF, like classical IVF, and then there's ICSI. That would be the 3. In insemination, we actually don't do the fertilization in the lab. The fertilization itself happens in the body of the woman. The only thing that the woman do is like a process and a stimulation to actually ovulate, and then when the woman is ovulating, we treat the sperm and try to process it in terms of to be the the best quality that you can have, and then just inseminate with that sperm, but the proper fecundation will happen in the body of the woman, so it's not like in vitro fertilization actually. And then there's IVF, like the classical one, and then ICSI. Normal, like classical IVF would be like we just put the eggs with all the cells, like when we take the eggs from the body um in the how it's called, in the egg retrieval, the proper egg has lots of cells surrounded. We don't remove those cells if it's classical IVF, and we just put those eggs in contact with the sperm that we have treated before. We always treat the sperm with gradients, and then it's the sperm itself who will fertilize those eggs. All right, and then you can have ICSI. In ICSI, what we do is we actually remove those cells. Removing those cells, we can see if the egg is mature or not. Only eggs that are mature can actually fertilize. If they are not mature, and there's no need of injecting them because they are not in the right stage of maturation, so they cannot be fertilized. So when they are mature, we can just check if they are mature or not, and then we just inject the sperm in the egg with a microscope. To inject the egg, it's quite kind of funny, I would say. What we need to do is we need to actually not cut the tail of the sperm, but we need to immobilize the sperm. So you will be like looking at the sperm that you have and trying to choose those ones that have like best motility and best morphology. And then if you would inject the proper sperm without actually, it's not cutting the tail, but it's kind of like if you have the tail, you just um smash it, kind of. I would not use that word, but in terms of you to check what I'm trying to say, and then the tail would stop moving, and that's because when you will inject the sperm in the egg, if the sperm would be still moving, the proper sperm will degenerate the egg because the tail will be like shaking, and it will be degenerate the rest of the egg. In the like in nature, the proper, when the sperm gets into the egg and introduces itself into the egg, the proper egg makes a mechanism, and the tail of the sperm stop moving. So we need to try to reproduce what's happening there and try to put the sperm as it would be in nature. So we actually need to stop the tail, and then you can see that there's a 90-degree angle of the tail when you have, it's not smash it, because smash it sound like if you are going to degenerate that, you're not going to degenerate that, but you need to make them stop moving, and then that will make like a procedure inside the sperm start. And that's important because if that doesn't happen, then the egg won't fertilize properly. That's it. That's like the three types of fertilization that we can have. Actually, just two, because insemination, it's not in the lab what we do it. But yeah. And then now this next question is about like conditions and temperatures that we need to have in the lab in order to not influence embryos development. Obviously, in the lab, we try to reproduce what's happening in the body. Okay, lab temperature would be always between 22 and 26 grades. That's the temperature in the lab. But then in the laminar flux cabin and then in the microscope, you will have heated surfaces that they need to be in certain temperature in terms of the eggs or embryo always be at 37 degrees. And then you will have the incubators. Inside the incubators, you will have certain air flux and also temperatures in terms of the embryos and eggs always be at 37 degrees. That's really important. It doesn't really, like, it depends on what dishes are you going to use or what media you are going to use. Then maybe you will need to have the heated surface at 38 or 37.5, wherever, but you need to be actually accurate and know that the media where the embryo will be, it will be at 37 degrees. We also try to work in a semi-sterilized lab. It needs to be as much as sterilized as you can have. Obviously, it's not 100% because we need to work with our hands, but it's as much as sterilized as you can. So you will use all dishes and everything that you're using in the lab. It's sterilized. Also, everything is single use, and that's pretty much it. About the temperature, for example, what I can say, there's some process that needs to be not in a heated surface. So we have a flux laminar cabin with heated surfaces and another one without. For example, freezing of the eggs and embryos, it's not in a heated surfaces. Cryoprotectors can never be in heated surfaces, but for all the rest of the process, we would work in heated surfaces. All right, even inside the like microscope where we inject and we do ICSI, the surface is heated. Okay. If you have any questions, you can ask or write something, or if not, you can always reach me by email. Okay. This it's quite odd to be talking here and answering questions without anyone kind of answering anything. I'll continue. Okay. Then the next question, obviously, it's quite related with that one. It's like, what's your workstation and what consists? Actually, I would say it's like a flux laminar cabin. That's what actually would be my workstation. With it has a microscope, and then you will have a microscope that it's called RIC, and there is where you would do the ICSI procedure, and where you will have like a higher amounts to see what the embryos or the eggs, and then the ones that you will have in the flux laminar cabin, it's like lower. You can have like a magnification, but not that much. I would say that's actually the workplace that we have. It's actually the lab, and the cabin. We actually spend most of our day over there, and that's it pretty much. You have a theater next to it, so the lab it has a door next to the theater. In terms when you're doing the egg retrieval, you will have, it's really fast to actually take all the fluids that the doctor will give to us in order for us to check if there's eggs or not. And when transfers happen, you actually, it's really fast to get there. So the lab will have a door, and next to that door there's the theater. But the lab itself, I would say it's actually the flux laminar cabin. That's it. That's where we spend most of our time. We have one cabin that it would be the cryopreservation part, and then the other one is where we do like the egg retrievals, transfers, and all the rest. The difference is because in one of them you will need the heated surface, and in the other one you won't, and that's pretty much it. Okay, and then there's one last question that's maybe a bit tricky. And it says, my lab decided not to do my IMSI as planned, just ICSI and IVF. Why might they not? All right. Um, I don't really know exactly your case and why that lab decided not to do that. You need a special microscope to do IMSI. And well, for anyone who doesn't actually know what's IMSI, IMSI it's kind of an ICSI, but you will have a better amounts of the sperm, so you will actually see much properly the morphology of the sperm, and you will be able to actually see more which one sperm is better in terms of morphology than the other one, to actually check and decide which one to choose to do the ICSI procedure. it's not that often that labs do IMSI. Actually, I think that I have worked maybe in one lab that actually does IMSI. It's not a really standardized process, and it's because it has its pros and its cons. I would say that EMC takes a lot of time, and in the lab you actually doesn't have that time to have the cells outside the incubators. It's not that you don't have time to do things; you obviously have the time, but you will need to try to have the cells, embryos, sperm, whatever, the least that you can outside the incubator, because it will be outside the proper temperature and pH and everything. All right. And obviously with IMSI you will actually be able to check properly sperm morphology. But if you have a sample that morphology is really really bad, IMSI actually won't help that much, because if the whole sample has a really bad morphology, you won't be really able to choose anything. Morphology will be bad anyway. And in samples that they have a borderline morphology, actually, you with ICSI cannot choose, you don't have as much as you can see in IMSI, but you can see a lot. Over there, obviously, with ICSI, you would choose what sperm is best in terms of morphology. And if the sample is like just borderline in terms of morphology, with ICSI, you would be actually able to choose and really kind of see which ones are better than the other ones. So maybe that's why your lab decided not to do that. I just think IMSI has its pros and its cons, and obviously, as every single technique that we have in the lab, it just doesn't need to be standardized. So it just needs to be chosen exactly which patient will actually beneficiate of that and which one no. And maybe if that sample that day of yours was good enough for do just only a proper ICSI, there's no need for you to pay for IMSI. There's no actual need for you for any extra cost of something if you can actually just have an ICSI. And that's it pretty much. We just don't have any more questions. That's everything. I don't know if I've talked quite fast or not, and if everything you can actually understand everything. I hope you did, and I hope you enjoyed these questions. It's quite nice to actually be able to be answering your questions. So whenever you have questions, you can reach me in our email. All right. And if not, just by Instagram, I'm sure they will send me your questions. All right. And that's it pretty much. It's a pleasure. I'm sure we'll see each other here any other day. And that's it. If anyone has any question, that's brilliant. All right. Bye bye.